Hello, everyone. Thank you for joining us for this virtual demonstration of the ion torrent Genexus system. My name is Andrew Hutchison. Please enjoy this short demonstration using our new virtual demo environment, after which I will answer your questions. Throughout the presentation, please submit your questions to the chat, and I'll get to as many as possible after the demo. This is the integrated ion torrent Genexus system. This unique system is the only true end-to-end NGS system available today from a single provider. The system automates all the key steps of the NGS workflow. At a high level, these steps include sample prep, library prep, template prep, sequencing, and data analysis. Before we dive into the workflow, I want to point out the variety of sample types that can be run using the Genexus system. These sources of genetic material include tissue, blood, plasma, and several other sample types used in oncology and inherited disease research applications. The Genexus supports a variety of clinical and translational research applications, including genomic profiling for oncology and hemato-oncology research, and SARS-CoV-2 genome sequencing. Let's get started at the beginning of the specimen to report workflow and focus on the initial steps of the Genexus purification system first. While it is certainly an option to run these units in standalone mode, the integrated configuration provides the most seamless end-to-end workflow experience. So let's review this option today. The process begins with creation of a sample to result run plan with which is appropriate for the intended assay. As depicted here, to load samples, simply remove the foil seal from the purification plate and pipette your pre-processed samples into the wells indicated by the run plan. Users then load consumables onto the Genexus purification instrument. As shown here, proper consumable placement is detected in real time to prevent setup errors. If no errors are detected, the run can begin. At the end of the purification run, a 96-well plate is produced that is ready for transfer to the Genexus integrated sequencer. Users then move the plate to the sequencer. And then you load the remainder of the sequencing reagents onto the deck of the system. And again, real-time feedback helps prevent any setup errors from being made. The final step before the run begins automatically is to place the bulk bottles and sequencing cartridge into the proper spots and close the doors of the system. You can now walk away. It's really that simple. At the end of the run, the instrument automatically decontaminates with UV lights and the onboard cleaning solutions. Simply come back when the run is complete to view results. The Genexus software automatically executes base calling, alignments, and variant calling. You can view run metrics and QC reports to check your run and data integrity. And the only thing remaining is to generate a report through your lab's reporting solution, like Oncomine Reporter. That's really all there is to it. The Genexus system is truly an end-to-end solution. You can generate next day results because of the highly automated nature of this system, with only 20 minutes total hands-on time. It's been my pleasure to showcase the Genexus purification system and the Genexus integrated sequencer for you today. If you are interested in trying a virtual demonstration yourself, this experience is available in the Genexus System virtual lab. Now, I'll be happy to answer any of your questions. So I'd like to actually remind everyone that on the last slide of that presentation, we did note that we do have support for new languages in that Genexus virtual environment. So if you were having trouble walking through the English version of that, there are more languages to walk you through it now. Let's get to some of the questions. All right, I see the first question here is, there have been a lot of new sequencing instruments released recently, including the Element Biosciences Aviti, the Singular Genomics G4, Illumina NovaSeq X, just to name a few. How does the Genexus compare to these? So indeed, it is a very exciting time for NGS right now. These instruments all have advantages in different applications, and the competition and technology advancements are great for the future of basic science and translational research. But the Genexus stands apart from the other instruments on the market in that it's designed to be extremely fast and easy to use. It's still the only NGS platform and specimen to report walkaway automation from one single vendor. And the Genexus is well suited for operators of all experience levels, research applications such as precision oncology, reproductive health, infectious disease, and inherited disease research. All right, next question. What is the cost to sequence a whole human genome on the Genexus? So we generally don't sequence whole human genomes on the Genexus. That's just not what it was built for. Many NGS instruments are developed for applications that require more sequence coverage, but lower sequencing depth, like whole genome sequencing. On the other hand, the Genexus was designed for targeted NGS and clinical research investigating smaller numbers of genes or genomic regions associated with various cancers or other diseases. In many cases, targeted NGS is the more economical, efficient, and effective approach. But in cases where whole human genome sequencing is required, other instruments that have higher throughput and less automation would be appropriate. Let's see, can I run libraries that I have already made for other ion-torrent instruments on the Genexus? So yes, you can. The Genexus supports library to result functionality in addition to the nucleic acid to result functionality. So if you have previously made libraries that you could run on previous systems like the Gene Studio S5 systems, you can indeed run those on the Genexus sequencer. Do I need to dilute my samples before placing it on the Genexus? So the beauty of using the Genexus purification system in integrated mode with the Genexus integrated sequencer is that the instruments do these steps of the NGS workflow with walkaway automation. After the nucleic acid extraction and quantification steps are completed, you simply move the NGS-ready plate from the Genexus purification system to that Genexus integrated sequencer and start the run. There's no additional pipetting steps that are required. Now, if you do decide to do either manual sample prep or automated extraction with a different instrument than the Genexus purification system, you do have to do some form of quantification, but you can just simply provide the concentration that you measured into the Genexus software, and you can allow the Genexus sequencer to do the onboard dilution for you. All right. Can I run multiple assays on the instrument at the same time? So yes, you can run multiple assays on a Genexus chip at the same time. There's 4 lanes, and you can run up to 4 assays, 1 assay per lane. The only caveats there is that you have to make sure you're running the same chemistry. So that would be AmpliSeq assays altogether, or AmpliSeq HD assays altogether. And those assays also have to share the same thermocycling profile for the library prep portion. How many samples can I sequence per run on the Genexus? So the Genexus allows you to automate up to 32 AmpliSeq reactions on a single run. Now, I say reactions because for several of our assays, they are 2-pool or even 3 or 4-pool assays. So with 32 reactions being automated on the instrument, you simply divide that by the number of pools in that assay to determine how many samples you can automate for nucleic acid to result on that instrument. Now, if you're not doing library prep on instrument and starting from already pre-made library, you can sequence more samples in a library to sequence run, and then you're just simply limited by the amount of throughput that you can get based on however many lanes of the GX5 chip you're running. All right, next question. How many reagent strips do I need for my run? So the Genexus consumables were built with a modular design such that you can scale the amount of consumables that you use per run to fit the number of samples that you're running or the batch size, as well as the amount of throughput you need. So we have several strips that support the library preparation portion of the workflow, as well as strips that support sequencing on each lane of the GX5 chip. Now, when you go to enter your samples and create your run and assign them to an assay, the software will actually calculate all the number of consumables needed. It will give you a printout, or you can follow the steps on the on-screen GUI, where it will show you how many of the strips to place on the system. And with the vision system, it will also indicate whether you place them in the right location, whether they actually are good to use or expired, whether they're appropriate for the protocol that you've selected. So it really helps to reduce the complexity of actually setting up the Genexus system. What read length does the Genexus support? So the Genexus system supports up to 400 base pair read lengths. Typically, most of the runs or assays that we have go to about a 200 base pair read length. But specifically for our myeloid assay or TCR beta, we do go up to 400 base pairs. See the last question here. How is the chip stored after usage if not all lanes were used? So because we have the 4 lanes on the GX5 chip, and you can select to use just one of them when you initialize the instrument, that would leave unused lanes of the chip, so 3 lanes left over. Now, after that first run, you have up to 2 weeks before you need to take that chip off. The chip remains on the instrument and it is ready to be used. We have stability on the instrument for the chips and any of the other buffers on the instrument for up to 2 weeks after that first run, after which we would recommend that you take them off and clean the instrument and reinitialize with new reagents. But that does give you 2 weeks to come back and use the remainder of those 3 lanes after the first run with 1 lane. All right, I don't see any other questions. So I think with that, I will close it out. Thank you for joining us at Oncomine World, and I hope you have a great rest of your day.